So my talk today is really not really delving into a lot of clinical practice, trying to give a picture that everything for the next 20, 30 years is going to be integrated with genetics and genomics knowledge. 50 years ago, we didn't have much of a clue about what this human genome is about. We make clinical diagnosis based on dysmorphology. We see a classical Down syndrome children or a classical Turner syndrome. We then put a compiling features of clinical features, and we say, oh, Down syndrome and Turner syndrome. When we were first able to look at the cells and look at the chromosomes, and we saw trisomy 21, everybody was excited. Well, we have a molecular diagnosis for this Down syndrome. That was the beginning of what is clinical genetics about it. Then came to what is called, with the light microscope, we can only look at the chromosomes, and now, but we know there are so many other syndromes and so many other children with dysmorphic features, we did not have a diagnosis. And this particular series of children have, you could see that there's a heart surgery done on the child, suggesting the child probably have congenital heart disease, and vice versa. And it actually, the next discovery was trying to enhance the diagnostic of this chromosomal analysis, and we were able to then diagnose a bunch of deletion syndromes. And this 22q deletion, or velocardiofacial syndrome, or otherwise called DiGeorge syndrome, it was a real discovery. Everybody was so excited. How we can diagnose with this, what is called FISH, or fluorescent in situ hybridization, where we can actually see the segments of chromosome. So when there's a chunk of chromosome missing, but the rest of the chromosome intact, we were able to diagnose this one there. But that was, already ancient. In 2003, the entire human genome was actually sequenced. When we sequenced a human genome, it was very, very simple message. The entire human genome was basically made of 4 base units. 4 base units, and multiplicity of these base units make up the entire genome. 3 million base pairs, A, C, T, or G, make up the human genome, and every human being is individualized, and there is even human live for another million years, still everybody is going to be different, because there's an infinite ways of putting these 3 billion base pairs into individual people. That is really amazing. So that makes you to come to the next conclusion or next hypothesis, how much of nature, which is how you are made up, that's genetics, or your genetic makeup, versus how much the environment or the external factors influence this one. For example, if you can argue malaria or some kind of infectious disease is not genetics, it's basically, it is an infection. But we will argue, not everybody get the malaria, get same outcome. Initially we thought, oh, it's a different metagenomics, that means the virulence of the bacteria or virus or parasite, or we will say a dosage of that. What we have now learned is that it's not that, it's the genetics or the genetic makeup. There are certain people are resistant to certain infection. They just cannot get it. There are certain people, the AIDS discovery was based on, there are certain people cannot be infected with AIDS virus. And then they found out these people actually have a polymorphism in the chemokine receptor that is responsible for the vaccine that is in development is going to happen. So every single, from infectious diseases to how your color change, your hair color change, everything is genetics. So from myocardial infarction to hypertension to diabetes to any common. So the genetics is something that, something is inevitable. Every medical students have to learn this in detail, because every single diagnosis and every single biomarker and every single diagnostic and management skills are all going to be based on what is your genetic makeup. Besides that, rather than taking a test, I think sequencing is already happening in the US. A newborn is born in the neonatal unit. The patient is sequenced, and it went through a big bioinformatics computer. It pulls out and exactly tell you whether this individual have a polymorphism. They are going to react to heparin. They are going to react to acetaminophen or any kind of pharmacogenomics, we call it. So we can avoid all these medication can be given. Or other aspect of this is that if the pharmacogenomics, the panel, the computer, supercomputer, will tell you what are the risk of particular medication is going to respond, or the chance of respond. If the chance of respond is only 10%, you're not going to waste this particular medication. So we don't have to go on giving one to other until the patient respond to a medication. This kind of a smart or not targeted therapy, or people call it is personalized therapy, is on the horizon already. I can take numerous examples of that. So the bottom line is to really convince you today that gene sequencing is the future. It is inevitable. It is going to happen. So might well as we better go ahead and learn about it and implement that in our day-to-day practice. And I am going to show you some of the examples of what is reality at the proof of principle. So I'm going to go through each topic's going to have about a couple of slides. So the basics, this is just for the medical students. I'm going to make this lecture series available for any students, so any of you want it, I will take some sensitive slides that have some face pictures on it there. And after that, I will ask the organizers to distribute the slides, so don't worry about taking notes, but just to go through that very quickly. So it's simple. I think it's a 46 chromosomes, is 22 pairs of autosomes and 1 pair of sex chromosomes. And this is what we used to know what is genetics about, is this chromosomal structure. I think what also we need to know a little bit about this genome. Genome can be DNA, RNA, or microRNA, or any kind of a genetic material is called genome. But DNA, which is now, as I say, is simple. It is based on 4 building blocks, 4 basic elements. It's adenine, thymine, cytosine, and guanine. It's put together in a different A or C or A or T, whatever it is. When you multiply this, that's what make it to a human genome. So it's not very complicated. So when we sequence and arrange everything in order, we can then individualize what is going on with individual people. So then comes to what is called genes. So although we have 30 billion these base pairs, each gene is responsible for human function. For example, how tall you are, or how short you are, or whether you are risk of, or you are going to develop cancer at the age of 50. Whatever it is, it actually imprinted in your genetics when you are born. Well, it can be modified. We are going to talk about this subsequently, whether gene silencing and epigenetics and methylation, that is different part, but at least we can agree that DNA actually you are hardwired when you are born with certain things. We know that. When a baby is born, obviously you could see the baby look like their parents. It's just coming from a DNA material that is imprinted. The same concept applies to multiple other factors. So the gene there are about 30,000 genes. Every function, everything is controlled by the genes. And then other simple thing we had to know about is what is called exomes. Only about 1 to 2% of this human genome is exomes. That means that code the protein. Rest of the region is called exons, which we used to call this a junk DNA, but not anymore, because now we know a lot of regulatory functions are done by that. Anyway, I'm not going to go through this, and I don't want to lose you on the lecture, but I think it is important to understand pieces of the DNA is responsible for most of the function. So the function is determined by whether you make the protein or not. There are certain instances protein are not made, certain instances of protein are made wrong. That causes the disease or dysfunction. Then the last word, I promise it's the last word before we go into clinical, is about mutation. So the variation, so mutation means it's not abnormal. Sometimes 40% of the population may have one particular mutation. That means that every autosome, everything have two pairs of that, and one come from a mother, one come from a father, and there is a major and a minor. So if it is 49 to 51, still that is mutation. So mutation is not always bad. It's just showing that it is a variation in the human genome. The mutation, we always think of mutation is harmful because we know that because that's where it is written, that's where it is published, but mutations are not always bad. Mutations can be arising spontaneously. That means it can be de novo. That means sometimes these changes can occur, but those are rare. On roughly about one mutation in a human can occur in one lifetime. So it's a very, very rare. So 99.999% of those mutations are coming from your parents and grandparents. That's where it is calculated. So I'm going to go through this quickly to what is called microarray. This is a complicated slide. I don't want to go through that one. But what is shown here is a very computerized mathematical technology, all automated. So you take a drop of blood, and the DNA is extracted automatically. It goes through a series of tests, and the supercomputer will spill out and stretch out the DNA molecule, and you can scan this part of the DNA where you are interested, and it can show you where this is about, the base pairs and what is the problem is. For example, sickle cell disease is a major problem in Africa. There's a one base pair dearrangement. Cystic fibrosis is a major disease in the Western world. Again, one base pair out of the 30 million, one base pair rearranged can be diagnosed, simple computerized technology. So quickly I'm going to go through what is single gene disorders and what is complex disorders about it. So simple gene disorders are usually made by one mutations or one deletions or one problem. When you have that mutations or the defect, you always have the disease. It can be autosomal dominant or recessive, X-linked, mitochondrial, or subsequently. Take an example of this one, this family. Both parents have achondroplasia, and it's autosomal dominant, but you could see that all three children were affected. We have known, because of short stature, we have known this disorder for many years, and now after these gene discoveries and genetic technologies, we were able to narrow down to one particular mutation in a gene called FGFR3. So it is fascinating to really go through the motion that we have known this disorder for so many years, And we didn't know the molecular mechanism or what is underlying etiology until now. Now we know the gene, numerous things can be done to either change or manipulate or whatever it is to maybe make those function differently for a different outcome. Neurofibromatosis is another one. It's a very common worldwide problem, and 1 in 3,000 individuals are affected with that. Now, actually, we know the defect or the mutation, but it's a 1 base pair difference, and we know the gene. Now, numerous pharmaceutical companies are interested in making drugs that can reverse the process as a gene therapy. So all of a sudden, these rare disorders ones are very interesting for pharmaceutical companies to make a case of it. Marfan syndrome, and one thing that I don't have time to go through that, but there are numerous discoveries and numerous advances are being made in Marfans to change their life. This is one thing I wanted to show you, what is called imprinting disorders. It is actually diagnosed when the fish became available or fluorescent in situ hybridization, where there's a deletion, a chunk of DNA is missing. But it became very interesting that if the deletion is caused by coming from the father or mother, one is Prader-Willi, one is Angelman syndrome. Both are totally different syndromes, but it is genetic defect is exactly the same. So it's the phenotype, that means what is the outcome of this is different. It's really clearly showing just a genetic defect doesn't tell you all that way. It also tells you all the way through from protein production to methylation to all the way there can be issues. So this is Angelman syndrome. I'm not going to go through because I'm going to allow these slides to be available for the students, and this all the features, and this Prader-Willi, which is also a major problem. But now we actually have a genetic defect known, and now multiple things can be done to look for it. So, there is a registry called OMIM. It is freely available. It is free for anybody. You look on this one. You can actually look for it. As of 2012, 21,000 single gene Mendelian disorders have been described in this MIM. But if you really look through that, because the gene discoveries and the sequencing technologies are just beginning to come, only about 10 or 15% of the single gene disorders are the molecular diagnosis or molecular mechanism is only known. Rest of them are still waiting to be discovered. I'm going to go to the next topic. So although it's fascinating, these syndromes and all kind of stuff, and because of single gene disorders, one gene defect causes the outcome, which is syndromes or disease. But 99% of what we talk about, including the lectures we talk about, for example, sepsis. Why certain people are susceptible to sepsis, certain people are not? Why certain people only go into they all go through a same infection or some kind of insult, and they go through a pre-stages, but they don't go into a severe sepsis and recover. And so now this hypothesis has now been tested, and we know there are certain mutations are responsible for the people who go through the severe sepsis. From infection to sepsis to cancer genetics is huge. So the common and complex disorders, including diabetes to heart disease and everything, is now caused by genetic mutations. So the Genome project have actually taught us a lot. Although individually we are 99.9% of the are similar, we share the same chromosome, that 0.1% of the chromosome matters that responsible for that. And other thing also we know is that there are about 30,000 genes. And most of the genes have, all the genes have some kind of a function. So again, we don't have to remember all these things because we have the supercomputers that can tells you everything for you, what is available. You just need to know how to, therefore, our laboratories and everything, half of the people that employ are actually not going to medical school. They actually have, they are mathematics, bioinformatics. That's what they do. They are engineers and they have PhDs in bioinformatics. They can assemble, deassemble these human genomes, and as we tell them this is a problem, this is a clinical history, they will narrow down everything because that's where they are trained for. So the Genome project is, I told you that is, it's the future, and this slide you can tell you is in fact, that is the future. So from disease susceptibility all the way to prognosis, genetics and genomes are going to tell you. So very soon, our older generations are very difficult to grapple this concept of we have to know this genetics and genomes. The medical students and junior doctors are going to tell you what to do because they are very quickly understanding this very rapidly. So we have to learn this. And again, this is a little bit intimidating how the sequencer put out your data. But if you really look very closely on that, it's basically 4 letters are repeated over and over again. Just 4 letters are repeated. That's what about the human genome is. So quickly going to the sequencing technology, there's one is called Sanger sequencing, where it took days and months to sequence certain part of the genome to what we have called NGS, or next generation sequencing, which is extremely cheap, and it is very cost effective, and it tells you within a couple of hours you can sequence one individual. So in the US, you can actually send to a company. You can send you a blood and a little bit, I don't know, $300 or something you send. They actually send you your entire profiles. It's a commercially available test. And they also tells you you have 30% risk of developing prostate cancer, and all kind of based on their readout. So when we see our patients in the clinic, they actually bring that readout to you. And so we are supposed to interpret this one. So it's a little scary, but we tell them it's still experimental, it's not there for prime time, but nevertheless, this is a reality we are facing, and very soon this reality is going to be worldwide, global issue. So we had to deal with that. We had to head on. So the way to do that is get ourselves educated and get on with the game and be on top of it. So I think this slide to show you how the cost is going down and down and down. 2 years ago, it's in the hundreds, but very soon it will be 5% or 10% of them, because these sequences are becoming. It's like when we had a first cell phone, it was the size of a brick, and now it's a smaller one. I mean, the same technology is evolving in the gene sequencing area as well. So this is the next generation sequencing, how they are going to show you everything. So the use of genetic data in healthcare and clinician focus is coming. It is the way. So rather than giving, this cartoon is showing you, patients are given certain names and numbers, and you scan with a cell phone, and that is gene therapy. That's exactly telling you this patient is swallowing a C. That's going to go and change whatever the dysfunction of that chromosome is about. So I'm going to skip this slide to go into this OMIM, or this database. So if you can look to see, it is given into autosomal, X-linked, Y-linked, and mitochondrial. So at the end, it will give you the total. So if you look at that, it's 22,000 total. But if you really look at the bottom part of the slides, it's showing you how many of them we already know the molecular diagnosis is. Not many. It's about 10% or 15% only we know. But nevertheless, it's really giving us so much fuel and so much excitement that we don't have to really give you descriptive diagnosis anymore. When the patients or the parents come to you, when their first child is affected, and they were devastated with their news, and they were going to have a second child. And what we used to tell them, we couldn't tell anything more than that and say, hey, it is genetics. If it's autosomal dominant, you have 50-50 chance. If it's autosomal recessive, you have 25% chance. That's not good enough anymore. I think we had to be now we can precisely tell, and then I'm going to show you some of the examples where we can actually change the outcome of this, not really aborting the fetus, but we can actually start the therapy before it's starting. So these are the examples of some human diseases, and the genes are known. And here I'll show you, not a lot of genes are not known yet, but we are on our way to find everything else. This is the common disorders. You could see it in the left side, PKU to cystic fibrosis, sickle cell anemia, to all the genes are now known. So all of a sudden, all the pharmaceutical companies and all the scientists, everybody is very interested, and US government is actually funding what is called a rare disease consortium. So even though the incidence or the incidence of this is 1 in 10,000, they are willing to fund to find it, because the examples of this lipitor or this cholesterol drug actually developed from a very rare disorder. And they found that it's a gene that is responsible for that, and they found that it's involved in cholesterol metabolism. So when they developed this small molecule, they actually found out that is actually going to be useful for wider variety of people with high cholesterol. So I'm going to take a case presentation, just a simple example. This patient presented with leukorrhea and severe feeding difficulty, Hirschsprung-like disorder, and hearing loss. And MRI shows cortical dysfunction. And this is a child is shown with the arrow, and the mother also was diagnosed with retinoblastoma at the age of 6 months. She had an eye taken out and no medical issues after that. And the brother had developmental delay, but no retinoblastoma. So, I mean, clearly this is, this is genetics, but we didn't know because with available technology, we didn't know anything. So we sent it for sequencing. And that's the fun. So the sequencing actually exactly tells you where the problem was with the single deletion in the family. This is another example of what is called Miller syndromes. We know about this, but we did not know the molecular diagnosis. And one patient and one family, father, mother, and affected child and unaffected siblings, we were able to diagnose the molecular diagnosis of Miller syndrome by just few days of this DNA sequencing. It was considered. So this is another example of what is called ocular cuteness, albinism, and neutropenia. And again, this was done through exome sequencing. This is another example of unknown mental retardation and the families. And then subsequently, this slide is a little complicated, but how we narrow down the diagnosis is all math, all mathematical modeling. There are algorithms that will fit into the computer, and with a few clicks of the button, it will narrow down to about 4 or 5. And we actually have well-trained human geneticists that they can help you to narrow down with the clinician. They will sit down with the clinician, and they can take it down. Again, this is a Charcot-Marie tooth disease. We know this is a occurrence, but we didn't know the molecular diagnosis until actually 2 years ago. We came out with the molecular diagnosis. We published this paper just recently about a patient who actually have FOXP3. The patient had severe inflammatory bowel disease, and we didn't know the diagnosis. You could see the mother was affected, and 3 boys all were affected. And we actually exome sequencing and able to narrow down the diagnosis. And it turned out to be it's a germline mutation. So this patient underwent allogeneic bone marrow transplantation, and the patient is completely cured. He does not have this bad inflammatory bowel disease anymore. So this is something that not necessarily a diagnostic interest, but also it can be completely therapeutics. And this is another example of a paper recently published that severe, these children will die otherwise. And now we have outcome can be altered by bone marrow transplantations. So the next thing that is coming is what is called these cancer syndromes. So we have now, because of the people are surviving more time and life expectancy increasing, the cancer syndromes, cancer diagnosis and molecular diagnosis are huge. A woman is diagnosed with breast cancer. It is just standard of care to go through them to BRCA or other genetic testing to define the therapy. That is already happening in every single woman. So your outcome is going to be completely different. If you have BRCA2 positive and they are recommending mastectomy as a primary modality, they are not going to go through radiation and chemotherapy anymore. So this kind of approach is already occurring very well. Cancer, I took the breast cancer, and the cancer syndrome, and this colon cancers are again very prevalent in our society. It is now becoming almost a reality. 2 slides on newborn screening. Yesterday we had a very collaborative meeting yesterday about a few things, and newborn screening come up. So why newborn screening? Because there's a skeptics tells about it's very rare, it's very costly, it's not worth saving a life when the disease occurrence is 1 in 3,000 or 1 in 10,000. In fact, again, this is another thing that is inevitable. It is a standard of care in the Western world. It actually, if you pick up these children before they actually present with the disease, you can prevent death, mental retardation, and subsequently all the morbidities. Besides that, you can actually give you genetic counseling for subsequent offsprings of these children. So this NBS on newborn screening, one panel, so when their babies are born, a blood drop is taken from the heel, and with this dried blood spot sent, so there's no tube, there's no cost associated with that. It's a very simple test. It is 31 disorders are diagnosed within 3 days of diagnosis. And this cost is very, very minimal because it's all worked out very well with that laboratories. So this is a very doable technology, at least starting very simple low-hanging fruits like hypothyroidism, hemoglobinopathies, and vice versa. Going back, in US, all the lysosomal storage diseases are also diagnosed now by newborn screening. So each state have a different panel based on their funding, but our state actually including these storage panels and stuff like that. So the other skeptics will also tell you, what can we do? Okay, you make a diagnosis, but what can we do? Actually, you can do a lot. Just simply limiting the diet or changing the diet. You can treat PKU, galactosemia, and medium chain fatty acids. I mean, galactosemia, you can prevent cataract, and you can prevent mental retardation, PKU. So the simple thing is provide treatment that can give you alternative pathways for removal of toxic, things like that. So I think the newborn screening is something something that can be again incorporated into our genetic panel and stuff like that. So rather than saying genetic disorders, I would like to say that everything from common diseases to rare diseases to infectious diseases to non-infectious diseases have implication for genetics. And because in other setups, we had to wait for the disease to occur, here we can actually prevent and institute therapy before the disease occurs. That's where the genetic disorders are going to be important. So again, this is a classical example. This patient have aniridia. So, but the patient is completely well. So this patient was watched, and patient then developed congenital cataract. And I'm not going to go through the whole story. And the patient had a renal ultrasound, MRE, endocrine evaluation, genetic testing, and we sent the sequencing, and it was clearly in the PAX6 gene sequence. And bottom line is this patient was given the appropriate treatment to deal with the problem, and the patient cancer was prevented. This is another example of what is called dilated cardiomyopathy. It's a very common problem in Southeast Asia. You could now see just not one gene. About 20 of the genes are contributing a small portion of it. In the past, when we are working like a silos, or when we are looking for each chromosome or sent for analysis of the certain disorder, we don't know how to deal with it. Now, with the sequencing, we can actually we can analyze and compute and assemble all genes discoveries. Next generation sequencing, not only a clinical tool as given in this slide, but is also a public health tool. So I'm going to talk about this little bit. 0.25% of the US women carry a mutation called BRCA. That is the lethal form of very early onset breast cancer gene. 85-time lifetime risk of breast cancer. 25 to 50% risk of ovarian cancer. So this knowledge will prevent ovarian cancer and breast cancer with appropriate screening or early detections. So next generation sequencing is actually can be a public health tool. Let's apply this to another common disorder in what we face in Sri Lanka. And that can be easily applied with the adequate technology. So I think it will come to the point when matchmaking come, and then she says, let's forget about it, and he's asking, is it in my DNA? And I think this is not a joke, but it's actually going to be a reality in future. So my talk is really about really giving you some proof of principle that what is the future is going to look like, and whether you like it or not, the technology is going to improve, the testing are going to be very easily available, and patients are going to do the testing. And not only a lot of lives can be saved, because we are not going to give some simple medication like heparin to the people who cannot tolerate heparin or tricyclics or whatever the problem is, all the drug-drug interactions. And so it's just not the pharmacogenomics. Everything is going to be on the genetics. So I'm going to stop here and then thank you.